Hello guys, welcome once again to another tutorial session on bioinformatics. And in today's video, I'm going to show you how to create Manhattan plots of FST values using Python. So before we go to the lab, let's quickly look at what the FST value is. There is a measure of variation among subpopulations and it is based on allele frequency differences. FST values are in the range of 0 to 1. A value of 0 indicates homogeneous subpopulations, whilst a value of 1 indicates distinct subpopulations. The workflow for this tutorial will be as follows. We will first get our SNP data, which is in the VCF format. And then using vcftools, the SNPs are going to be filtered, FST values calculated, and then the bioinfokit library in Python will be used to create the Manhattan plot. The script for this tutorial is available on my GitHub page, and so you can always go and then download them. And to be able to follow this tutorial, you will need to have a Unix environment on which is installed Python 3.6 or higher, pip3, virtualenv, git. Automatic installation will be done by the script for vcftools, mock, and then other Python libraries which will be needed. Ladies and gentlemen, let the analysis begin. So first of all, you need to open your terminal and then we will download the repository. Okay, so you can download using the Git, or you can download as a zip file and then extract. But I'm using the Git to do the download. So after download or cloning of the repository, you can confirm with our ls command. And so the directory would have been created. So we will cd to that directory. And in that directory, we can do an ls to find out what is there. So we have some scripts here. The next step is to add execution rights to this script that we are going to use. And so we'll do that using this command. Okay. I didn't bring the X. This is how it's supposed to be. Okay. So after that, if you are using Ubuntu and you do an ls, then you see that the color of the script files has been changed, indicating they are ready to be executed. So we can proceed. The next step is to download the data. Okay, and to download the data, we will use the getData.sh to get the data. So we wait whilst the data gets downloaded. So the download is complete. So let's quickly look at what has been downloaded. And that is the Manhattan plots data zip file, this one. And so we need to unzip it. So after unzipping, when you do an ls, you will find there has been a new directory created, data, and that contains what we will need for the analysis. Okay. So a little bit of info about the data. This is a Plasmodium falciparum data that I got from the malaria gene database, the PFK project. Okay, so we have So we have the snip data here, and then we also have this text file, So this text file contains sample IDs for Plasmodium falciparum isolates in Bangladesh, and this one contains isolate IDs for Gambia. Okay, so now we have Plasmodium falciparum SNPs for two populations, Gambia, Bangladesh. And so that is what we will use in the downstream analysis. Okay, so now let's look at the next part. So the next part is to download the tools. Okay, so we do that using the download tools script. So this will download, configure, and install tools like vcftools, mock, and then other Python libraries for us. And because we are compiling them from source, to take some time before everything is set. So now the download and installation of the tools has been completed, and you find this text displaying indicating what commands to run next. Okay, so we will run these commands next. So this first command adds the tools to the path so that you can call them anywhere you are. Then this one will test the installations to see that it's working. So let's do that. So source. Once you do that, you find that this name has been added, indicating a new environment has been created and activated. So now we will test the tools. So the testing will display the versions of the software tools that are going to be used for the analysis. Okay, so we have vcftools, mock, Python, and also have versions of Python libraries that we are going to use. So that is for the software installation. After the software installation, we are now ready to start the analysis. And we begin by doing the filtering. So with the filtering, we will use the raw data, SNP data, and then perform series of activities on it to ensure that we have high quality SNP that will be used for the analysis. Okay, so to do the filtering, we call on the filter snps.sh, and then we specify the SNP that we want to filter. That's the SNPs index. And then we proceed with the filtering. You might see this warning being written by vcftools. Okay, this warning can be safely ignored. Basically, what it's indicating is that it can't handle the comma characters that has been introduced in some of the fields. Okay, this is because the raw SNP data, the SNP data was annotated, okay, using a tool, and that particular tool added this description, which includes this comma characters. So that is what this warning error is. So the filtering has been completed. Okay, so after it has been completed, we are going to find some files being generated. Okay, so if you do the ls, you find these files here, which are intermediate files that we don't need. But this is the final file that we are going to use, the filtered data. Okay, so this is what we'll use for the next analysis. So let's clear the screen again to make it nicer, and then we proceed. So after filtering, the next stage is to now calculate the FST values. Okay. You use the .get fst.sh. Now, with the FST values, you need the populations that you want to do the comparison on. That's the two populations, and you also need the SNP data, which is in the VCF format. Okay, we know our populations to be Bangladesh isolates and then Gambia isolates, and this they need the IDs are in this text file, so we specify these text files also as inputs. Okay, so now we will do get FST, and then we specify the two population. Okay, once we are done with the feeding of the populations, then we now specify the SNP data. Okay, we are using the SNPs.filtered. That's the filtered ones that we got. Okay, so we proceed to do the computation, and it gets completed, and you have this also shown. Okay, so we are going to have this file generated, the log file, and then the file that contains the FST values, which is here. So this is the file that we are going to use in the next analysis. But let's quickly look at what this file holds. So the FST values will be calculated for each chromosome and then the position. Okay, so this is and how it's going to be saved for us. Now on the Manhattan plot generation, the name of this chromosome will be absent, but rather we replace it with a single value. This is because the script numbers the chromosomes and then uses those numbers to do the plots. And so you have chromosome 1 to 14 on the plots instead of having these chromosome numbers. Okay, so this is how the FST is generated. Okay, so now we generate the Manhattan plots. Let me clear the screen again. So we generate the Manhattan plots with the plots.fstpy. Okay, so this particular file contains commands that will generate the Manhattan plots. Okay, this is the main file that will do the plotting, but this will serve as intermediary file which will just pass on some information here to do the plotting. So we call Python 3, and then we indicate the file, and then we also push in the FST values, which is saved with this name. Okay, so now we are ready to plot. So within some seconds, the plot will be generated for us. Okay, with the indication of this message. So if you should do an ls, you will find that a new PNG file has been created. That is the Manhattan plot. Of course, you can also view it graphically without the command line. Okay, so for me, because I was working from the desktop, I can actually go there and then find out. Okay, to find where you are in the directory, you can just issue pwd. Okay, and that will give you where you are. So this is my home directory, and then this is my other directory. Okay, so in this directory, this is where I have the Manhattan plots generated. So I'll quickly go there and then we'll just have a look at it. So this is my desktop, and this is the Manhattan plots that has been generated. You can see it here, okay, as this particular file. Now let's look at how we are going to interpret the Manhattan plots that has been generated for the FST values. This is the Manhattan plot that was generated for us. So you have the chromosome numbers, and they also have the FST values, which is in the range of 0 to 1. Okay. Now the Python script I'm using also allows you to change the colors for the plots. Okay, so you can have another color like this. So depending on your preference, you can choose any color combination that you want. Now for the Manhattan plot that has been generated for FST values, you can also set a threshold for the FST values. So for example, if you should set a threshold of 0.5 here, then what this means is that SNPs here that are above this particular threshold line can be considered as selection signatures. Okay, so the genes on which these SNPs occur might be associated with certain genetic traits. That is what this FST value lets us know. And as we see, 0.5, we are gradually going up to 1. And so selection signatures will indicate what features makes this population distinct from each other. Okay, so this is how I bring my tutorial to an end for the Manhattan plot. And I hope you get to practice and then have your own plots. Have fun, then have a nice day. Bye-bye.